Welcome to the muscular system. There are 4 types of tissue, as you learned in the tissue chapter, and muscle tissue is specialized for contraction. There are 3 types: skeletal, cardiac, and smooth. Visually, they look different. Skeletal muscle has long cells that are called fibers and is multinucleated. Cardiac muscle tissue is branched, and you can see these odd things here. They are connections from one cardiac muscle cell to the next. It's called an intercalated disc. These are uninucleated. And smooth muscle tissue, which often occurs in sheets, is uninucleated, and the cells are called spindle-shaped. Skeletal muscles move the body. Cardiac muscle is in the heart, and smooth muscle is found in internal organs to regulate movement along those organs, as well as regulate the flow of blood through arteries. Skeletal muscle produces movement, maintains body posture and body position, and supports soft tissue. If you look at the pelvic cavity, at the base of that pelvic cavity is a large opening through which the infant's head passes when it's born. There is no bone there. It is held by muscle tissue, which supports all of the abdominal viscera. Muscles guard entrances and exits using sphincters, round muscles. Muscles maintain our body temperature because they produce heat as the muscles contract, and muscles provide nutrient reserves. We can use our muscles as energy if we don't get enough calories from carbohydrates and lipids. So we would break down our skeletal muscles to keep us alive. But also within the muscle will be glucose in its storage form, glycogen. The connective tissue layers of muscle are important. The outermost connective tissue layer is called the epimysium. The middle layer is the perimysium, and the perimysium will surround bundles of fascicles. So here's a picture. Here's our whole muscle. So it's hard to understand some of these pictures, so be careful of what you're looking at. Make sure you're looking at a muscle or a muscle fiber. It's kind of hard to distinguish. So here on the left, we are looking at a complete muscle, and surrounding that muscle is the epimysium. The muscle is subdivided into units called fascicles, and each fascicle is surrounded by perimysium. And then here's an enlargement of the perimysium. It is composed of cells. So here a muscle fiber is a cell, and that muscle fiber is surrounded by endomysium. So the innermost layer is the endo, and the outermost layer is the epi. So these collagen fibers extend throughout the muscle tissue and extend beyond the muscle tissue to become tendon. Now, sometimes these tendons are actually sheets of tendon, and that's called an aponeurosis. For example, we have one on the top of our head that connects the muscle of our forehead to the muscle covering our occipital bone. Different terminology in this chapter. A cell is a fiber. The membrane is a sarcolemma. The cytoplasm is called sarcoplasm, and the endoplasmic reticulum is called sarcoplasmic reticulum, and that is a reservoir for calcium. These skeletal muscles are multinucleated with hundreds of nuclei. They're filled with structures called myofibrils that are packed with myofilaments. These myofilaments are composed of thin and thick proteins. The thin protein or thin filament is actin, and the thick one is myosin. It is these actin and myosin filaments that give the striped appearance of skeletal muscle called striations. So here you can see these striations or stripes. Which of the following surrounds an entire muscle? That is, what is the outermost covering? Epimysium. Which of the following units is the smallest? Myofilament. The sarcomere is the functional unit of skeletal muscle. There are about 10,000 per myofibril, so gazillions per muscle. And here are the different parts of that sarcomere. We do not need to learn all of these different parts. One of them is called the Z-line, and that is the end of the sarcomere. So here we see two complete sarcomeres. One sarcomere goes from this Z-line to this Z-line, and then there's a Z-line right here. So it is the Z-line, this zigzag line that is at the end of the sarcomere, that is called the Z-line. You will find that there are many other proteins involved in muscle contraction, but the two that we are going to talk about most will be the thin filament actin and the thick filament myosin. I will give you a little detail about tropomyosin and troponin, but you will not be tested on it. So here's our thin filament actin. And you can see that the Z-line is composed of actin, and these thin filaments extend from the Z-line into the middle of the sarcomere. So here is our actin. There are what are called active sites on the actin that are covered up with a protein. Oops, what happened there? With a protein called tropomyosin. See this blue chain here? It's a protein. It's covering up the active sites. It's kind of locked into position with troponin. Well, you've got to unlock those active sites in order for a muscle to contract. And the key to unlock those sites is calcium. So we're going to look a little bit at the myosin molecules. They have what are called heads, and these heads can move. So here is a myosin molecule. Think purple in all of your diagrams. This structure here that's purple consists of myosin. The actin filaments go in between the myosin filaments. So here's the middle, right down the middle of the sarcomere, and here are many, many myosin molecules, and each myosin molecule has got heads attached to it. See these heads? These heads can move. Look, there's a hinge there, and the myosin head can flip. A membrane potential is just being defined here. A membrane potential means there are charges on each side of the muscle fiber membrane. That is, what provides these charges? Ions. Ions are positive or negative. And when you have a membrane potential, you've got positive and negative charges on opposite sides of that membrane. It happens to be negative inside and positive outside, but we'll see that a little bit later. When there is an action potential, which can occur in a neuron or a muscle cell, when there's an action potential, it travels along the sarcolemma or the membrane. And it's going to enter the cell by a structure called a T-tubule. So here's our cell, here's our sarcolemma, the membrane. The blue here is Sarcoplasmic Reticulum, which is a calcium reservoir. So just imagine a muscle impulse or action potential traveling along the membrane here. It gets into the cell through what's called a T-tubule. So this action potential travels throughout the cell by these T-tubules. The purpose of this is to get that action potential through the entire cell instantaneously, and it causes the release of calcium from the sarcoplasmic reticulum. This is describing those transverse tubules. It's continuous with the membrane, the sarcolemma, and it forms passageways through the muscle fiber. The sarcoplasmic reticulum itself is a calcium ion reservoir. It surrounds each myofibril, and therefore it really fills the entire muscle cell. So at the beginning of a muscle contraction, there's some sort of change in that membrane potential, and it causes this membrane potential to travel down the T-tubule and go to the sarcoplasmic reticulum to let the calcium out. Which part of the sarcomere anchors the thin myofilaments? The thin ones are actin, and that would be the Z line, the only one we need to know. So how does a muscle contract? It has to have an impulse from a neuron. So an action potential or a nerve impulse travels down a motor neuron. We have two types, basically two main categories of neurons, motor neurons and sensory neurons. And motor neurons carry information out to muscles and glands. So there's some kind of nerve impulse traveling out the motor neuron, and it's going to travel down the axon to the muscle. Now, this connection between the neuron and the muscle is very, very important. It's called the neuromuscular junction. The neuromuscular junction is where the axon is near the motor end plate on a skeletal muscle fiber. This region where the axon terminal approaches the muscle causes that muscle fiber to modify itself somewhat, and that modification, that whole region where the axon terminal comes down to the muscle, is called the motor end plate. Inside the axon terminal will be vesicles filled with a neurotransmitter called acetylcholine. And acetylcholine is crucial for muscle contraction. Now, not only is acetylcholine crucial, but the enzyme that breaks it down is crucial. The enzyme that breaks it down is called acetylcholine esterase. And that's found in the synaptic cleft, or the gap between the neuron and the muscle fiber. So that action potential travels down the motor neuron to the end and causes acetylcholine to be released into the synaptic cleft. This process is called exocytosis. And then the acetylcholine will cross that synaptic cleft and bind to receptors on the other side on the motor end plate of the muscle fiber. When it binds, it's going to open up the muscle fiber to sodium. Sodium will rush right in. And rushing of sodium into the muscle fiber generates the action potential on the muscle fiber. This would continue, except that once an action potential is generated, that acetylcholine is broken down by the acetylcholine esterase. And so you'd need another action potential coming down the neuron to have another contraction. Now, the action potential that's generated will cross the entire sarcolemma almost instantaneously and